Hi, I'm Matt Newton from the Marine Directorate. I'm going to give a brief overview today of some data that we have collected from an expansion of the West Coast tracking project, which has been looking at tracking salmon smolts up at the West Coast into the Atlantic Ocean. Over the last few years, there's been a large tracking project on the West Coast, primarily run by the Atlantic Salmon Trust, and their website is down at the bottom here, which has a really good overview and some nice infographics of the project itself. It's a large collaborative project funded by many different mechanisms over multiple years, as I said, led mainly by the Atlantic Salmon Trust. In 2023, they tagged 1,100 smolts from 12 different rivers and primarily focused on the sea lochs. And there's a few examples here on the right of the red stars of where fish were tagged and released. And in previous years, some of the yellow spots is where they've had receivers for deployments. But clearly this provided a great opportunity to provide some added value, and what we were able to do through the Marine Directorate and some of the ScotMER funding was purchase some extra tags for the project and deploy 21 receivers across the north coast out into the open ocean. The 21 receivers spanned approximately 120 kilometres across the north coast here. We struggled to go any further west due to the amount of water depth and that it makes it more difficult to anchor in more pieces of equipment in this location, and again also to the east, fairly constrained by fishing activity here that we want to be able to deploy our equipment and get it back. The acoustic receivers, this was designed as a leaky line, so there was potential for tagged fish to pass through the array without being detected, and there was a compromise between being able to cover the amount of ocean that we have, so the 120 kilometres, and the detectability of the amount of fish. What's really important, this is an area of unknown information. We've never tracked fish or detected salmon this far north of fish that have been tagged previously, so it's going to provide us with some novel results. Our general hypothesis was that we would see fish briefly as they migrated through the coastline up out into the North Sea, and on their migration towards the Norwegian coast, the Norwegian Sea, towards the north, and that we'd probably expect that most of these fish detections that we would see would be constrained to the west, so more on this side of the array as the fish were heading north, potentially looking at using the Atlantic shelf currents to get further north in a more energy efficient way. We detected a total of 72 salmon smolts. 33 of those came from the Laxford, 16 from the Snizort, and some from the Laxey and the Grimster, and also one from the Loy and the Lundy further to the south. What was really interesting is that we had far more detections than we anticipated, although it's not many of the 1,100 fish from the rivers that we did get. So for example, 33 from the Laxford represents about a third of the fish that were tagged in that population. So that's quite a lot of detections, given that this was a leaky line, it wasn't connected to shore, and fish would be able to pass undetected. The detections were spread across the entire array, and also quite a few to the east, which was quite surprising. And if we look down at specific populations, we can see that in more detail. So fish from the Laxford, many are detected over in this region here, and the reason why we have this sharp kink is these lines are just hypothetical, and we've used these for visual representation, so they're not allowed to pass closer than 5 nautical miles to the coast, hence why this dotted line is here. These lines coming out just around this 5 nautical mile buffer that we have made artificially. We have no information on how fish actually travelled from the river itself out to where they were detected on this line. This is just a hypothetical line itself. And similar from the Snizort, a few fish detected to the west, but quite a few fish detected a long way to the east of this line, right across to the end where those fish were detected. So those fish appear to be coming quite a long way east than we might expect. What showed most interesting information was the total time that fish spent on the marine array. So this is the time it takes from the first detection to the last detection, and we had some fish that were detected on that array for up to 15 days, as we see by the graph here. Each of these bars represents a single day, and it is the total time that each individual fish stayed within that array. So if you remember, our hypothesis kind of expected the fish would pass through fairly quick, probably in the first couple of bars here, 1 to 2 days as they headed north, but we had quite a number of fish, probably up to 25%, maybe slightly higher, spending a significant amount of time on the marine array itself, and indeed a number of fish up to 14 and 15 days that they were detected there. And it was the same across populations, so it was not a single population that was showing this behaviour, it was across all populations. And you can see here that from each river there are a number of fish that stayed on that marine array and within that area for quite a considerable amount of time that we might assume. And that wasn't as expected. We expected fish should be heading north relatively fast. Reasons for this behaviour. The first one obviously is predation. Some of the tags that we had have depth and temperature on them, and we're able to examine these to see if they were normal. So were there any hot tags indicating that a fish might have been predated by a marine mammal, or the fish swimming particularly deep that we might not expect from a smolt. First one here at the top left, this fish was actually detected on the array for a total of 13 days, generally in the surface waters down to about 4 meters deep, the temperature about 14 to 15 degrees as we'd expect from the sea water temperature at this time of year. And same for the other fish where we had these scent tags, 5.5 days, 4 days. I think this is also 4 days. Depth is all roughly around the surface as we'd expect. Temperatures all around about 14 degrees, again showing very little indication of predation or unusual behaviour. What we have seen here, and we see this on some other fish well, is there's potential diving behaviour, that the fish are not just utilising one part of the water column, that they're using multiple parts of the water column within the relative surface area itself. So this fish here has gone down to 10 to 11 meters in depth and back up again. And you can see that in the temperature as well. As they get deeper, it gets slightly colder, which is somewhat interesting. And moving on to this, that beginning to question, is this a milling behaviour? Is it a feeding behaviour? Why are these fish staying around for so long? And so looking at the graphs here, M1 represents the furthest east receiver and M29 represents the furthest west receiver. Between each of these receivers there's about 3 nautical miles. And so what we see is that the fish are not specifically moving along a line. And by what I mean from that is that they hit a single point and then they move east or west like this. So like this fish has here. The majority of fish are showing patterns like these, where they are back and forth between different receivers over not a very far distance. So for example, these detections here are only 3 nautical miles apart and the fish is going back and forth as opposed to moving along the line itself. And that's the same for this fish as well. As I said, the majority of fish are showing this pattern, as though it's a milling behaviour, potentially feeding. We're not quite sure, but there is not an indication that this is a directed or a strong migratory movement from these individuals. As you can see from the depth use of fish that had tags in them, during the day the fish were much deeper by these red dots or pinky dots, on average about 7.5 meters deep and also quite varied, whereas at night the fish were predominantly in the surface layers around about 4.1 meters deep, so much shallower and less varied. And it might be interesting to discuss this at the end about why this behaviour was seen. And finally, coming on to some conclusions, the array detected many fish from different populations, and those detections were spread across the entire line, including to the east. There was potential evidence of some milling behaviour or some non-directed migration. There was potentially feeding, and the depth use between day and night was considerably different.
Hello, my name is Hannele Honkanen, and today I will be presenting a high-level introduction on a piece of work that my colleagues and I have very recently completed for the Marine Directorate. This was a literature review on diadromous fish in the context of offshore wind. As we know, with the ongoing climate change crisis, there is an increased need for renewable energy proposals, and particularly offshore wind. Scotland's goals include plans to achieve net zero carbon emissions by 2045, and development of offshore wind will play a large role in this. While offshore wind has many benefits, there are also potential negative impacts on the surrounding habitats and fauna, which may also include diadromous fish of conservation concern. So there needs to be a balance to consider both of these and make sure that the potential impacts of any future offshore wind proposals are carefully considered. So the overall aim of this project was to review the current knowledge and provide recommendations for future research in relation to the relatively unknown interactions between diadromous fish in the context of offshore wind energy development. More specifically, the project had 3 aims. 1, review available information on distribution, movement, and abundance of diadromous fish at sea. 2, review available evidence to identify potential impacts resulting from marine renewables on diadromous fish. And 3, identify and prioritize future research and monitoring to address evidence gaps identified in aims 1 and 2. This review focused on 10 diadromous UK fish species, that may be found in Scottish marine waters. These were Atlantic salmon, the Diadromous form of brown trout, river lamprey, sea lamprey, three-spined stickleback, European eel, European flounder, European smelt, Alice shad, and thwaite shad. Diadromous fish have complex life histories, and most of them are of high conservation value. These 10 species include SAC qualifying species, species found in the Habitats Directive, and one species, the eel, on the IUCN Red List. The work for this report was done through a literature review covering both academic and grey literature and a series of expert panels. We ran 5 expert panels, each focusing on a different topic. One focused on the distribution of these diadromous fish in Scottish waters, and 4 on the potential impacts of offshore wind. Experts for each panel were identified through authorship in key papers, and this included experts from both academic and government backgrounds. As mentioned, the geographic focus was on Scottish waters, and especially investigating the potential overlap of diadromous fish and plan option areas for the 15 sectoral marine plan for offshore wind energy and 13 innovation and targeted oil and gas areas, shown in the map here. The key finding for this aim was that currently we really do not know much at all about the marine space use of these species around Scotland, and therefore it is really difficult to assess the potential overlap with high confidence. And this is really one of the key bits of information that we need, because all the work and assessment that follows will be based on this. Not surprisingly, the most information available is for salmonids, and especially Atlantic salmon. For salmon, we have some very good tracking studies providing us with actual migration routes and temporal data as well. So we know when they are migrating and how long these fish are spending in these areas. Considering the available data and expert panel feedback, the consensus was that it was very likely that Atlantic salmon are, either as smolts or adults, very likely to overlap with all of these plan option areas. On the other hand, practically no information is available for river lamprey, sea lamprey, smelt, and the 2 shad species. It is possible that these species will overlap with at least some of the plan option areas. However, we just do not have the data to be confident about it, and that is concerning. The lack of tracking studies also means that we do not have data on actual migration routes for many of the species. So we don't know if individuals and populations are overlapping with multiple POAs, which could lead to cumulative impacts. And additionally, apart from tracking studies where it is well known which populations these fish originate from, other types of data, such as trawl data, generally will not provide information on the origin of these fish unless genetic work is done, therefore making it impossible to assess exactly which populations are being impacted. And some populations may be more vulnerable than others, for example, the SAC populations. As part of the synthesis, we identified 4 key topics for potential impacts of offshore wind farms on diadromous fish. These were sound, electromagnetic fields, changes in light patterns, which includes shadow, flicker from the moving turbine blades, and light reflection of the surface of the blades, and issues associated with novel habitat creation, which includes community change, predation risk, suspended sediment, vessel activity, potential increased disease risk. This is not a comprehensive list, but we consider these to be the main issues. In the report, for each of these impacts, we have provided an introduction, review of available evidence, the list of species-specific research where that was available, summary of the expert panel workshop, and our assessment of the impact. There is not enough time to go through the findings of this aim in detail, so please refer to the full report once it's published, and I will just highlight some of the main points here. Again, for all the topics, there are clear evidence gaps, and overall there is relatively little available research, especially for diadromous fish species. And because of the huge variety in the anatomy and physiology of different fish species, it makes it quite difficult to extrapolate from the existing studies. Therefore, we currently are in a situation where we cannot estimate the potential impacts with confidence. However, it is likely that there are some negative impacts, and these potential impacts include changes in behavior, such as avoidance, which could be short or long-term, changes in physiology, such as hormone levels, physical injury, such as barotrauma, issues with navigational cues, and even death, although that might be unlikely. The likelihood and magnitude of these impacts will be linked to the encounter rate and amount of time spent by the species in and around wind farms, which is likely to vary between species. When assessing the existing research, there are a few points to highlight. To build a complete picture of the potential impacts, both lab and field studies are needed, and the different life stages should be considered, because what may not be a significant impact for an adult fish could be a severe one for a larva stage. To conclude, our recommendation is that the key research focus should be establishing the marine space use and therefore the potential overlap of diadromous fish and planned or existing offshore developments. Other key areas of research when it comes to the potential impacts of offshore wind, we suggest targeted research on the impacts of sound, electromagnetic fields, and potential changes in the predator-prey interactions. And interdisciplinary research is needed, especially when investigating sound and electromagnetic fields. We provided some examples of suggested research topics in the report. Thank you for listening.
Hello, my name's Adam Piper from the Zoological Society of London. I'm going to talk to you about some studies we've been doing to try and identify the migration routes of salmon post-smolts and look at a potential overlap with the offshore wind energy development in the North Moray Firth. This map provides an overview of the study array. Each pink ring is the location of a receiver. The black crosses are wind turbines. The green perimeters are showing you on the left hand side the Beatrice site, and on the right hand side the Moray East wind farm site, and the yellow dot is where we capture and release the study fish. Just to zoom in on the bottom of the wick there, those pink rings again indicate the receiver locations. That's just to give more high resolution information of when they leave the freshwater area and migrate out into the Moray Firth. So the study aims to provide an evidence base for possible impacts of offshore wind development on emigrating salmon. This is information that's desperately needed to aid the planning and consenting of these developments. We had a couple of working hypotheses. The first is that smolts would swim directly north, which would be their shortest route to their likely feeding grounds. Alternatively, they may initially follow the dominant southeast flow currents known to exist in the Moray Firth. This could help minimise the swimming energy expenditure. As the smolts are migrating downstream on the River Wick, we capture them in a box trap, which you'll see in the bottom left hand of the screen there. We managed to capture 108 smolts between the 13th and 22nd of April in 2023, and depending on the smolt size, we tag with either a V6 or a V7 acoustic tag. You'll see there that most of the fish we caught were between 120 and 145 mil, but there's a few bigger fish in there as well. So we'll capture the fish, inspect them, make sure that they've got no signs of obvious damage or disease, and then we'll anaesthetize them, internally tag them, and then release them. We usually wait till early evening time, till lower light conditions to release them, as they're less vulnerable to predators at that time. On average, fish took around 6 days to travel the 11.1 kilometres from their freshwater release site down to the marine environment of Wick Bay, so that's crossing that first black dotted line on the left hand side of the screen. A total of 95 fish, which is 88% of all those tagged, reached Wick Bay, and that reflects a 1.1% loss rate per freshwater kilometre, which is comparable with similar studies. And then further, one fish was lost in the Wick Bay itself, so that's between the two sets of black dotted lines. This graph's just to illustrate that most fish after leaving the river pass through Wick Bay quickly. So you'll see the majority of fish pass through the area in less than 2 hours. Certainly by the time it's 4 to 5 hours after first being detected in there, most fish had moved out into the Moray Firth. We see on these two plots that fish are both first detected leaving the river and departing Wick Bay in low light periods at the end of the day going into dusk, and of the 94 fish that emigrated to sea, 39 did so on an ebbing tide, with 55 doing so on a flood tide. It's important to make the point that we only have data back so far from the receivers that are marked black there. All of the receiver locations that are in grey are ones that are still out there but we haven't downloaded yet. So it's just to emphasise that what I'm going to be presenting today is just partial data, so it represents a minimum rather than final results. Despite not having data from the full array, we've already detected 34 smolts. You'll see on the top line of data there that their first marine detection, we've detected the majority on the north line, as we might expect, as they're coming directly out of the wick there, but also 9 on the south line and 5 within the turbines. And then for their last marine detection, which is indicative of the route that they were heading, I suppose we've got 18 on the north line, but still 7 on the south line heading in that direction, and 9 going through the turbine array. Here we're looking at detection split into zones. That's the number of unique individuals detected in each zone. That's really just indicative of the areas that fish visited, and you'll see here that on the northern and the southern line we've split the receiver array into inner, central and outer, indicated by the red, green and blue, and then the wind turbines all as one area. And we see the majority of fish are visiting that northern line there, but we're all still getting a substantial number coming down onto the south line and into the turbine area itself. We saw the majority of post-smolts move through the marine array relatively quickly, with 71% spending less than a couple of hours actually being detected within that array. We're already seeing a lot of interesting individual fish movements coming out of even this partial data. I'm just going to share a couple of examples with you. So this fish comes out of the River Wick and is detected first on the outer part of the northern line, moves down the line being detected, moves into the northern end of the Beatrice site there, and then moves across into the Moray East turbine site. This second example is a fish that appears to move south initially before moving along the line a little and then swimming north, then appears to turn and head south again. And we don't know what happens to the fish after that, so it'll be interesting to see if it's detected on the Beatrice array once we get the rest of that data back. To summarise findings so far, we know that a minimum of 36% of the fish didn't head directly north. Of these, most of the migration paths had potential to interact with either the current or proposed wind farm sites. The time actually spent within the turbine array appeared to be quite short, but I emphasise that it's incomplete data. And then we observed these complex south and north and sometimes south again movements of some individuals. So instead of these direct paths we might expect to see, this means that individuals have got potential to be exposed to turbine array areas multiple times. So looking at future work, Marine Scotland plan to download the rest of the receivers early this year, which will be great for filling in those data gaps. There's going to be a second year of the post-smolt tracking to build up that data set. We're hoping to do some sea trout translocation work. Now that's to look at the movement of tagged sea trout both within the wind turbine array and compare it to a parallel control zone. All of the tags that we use this year will have depth and temperature sensors. That'll be great for understanding the behaviour around turbines and may indicate particular events like predation events. And we hope to look at our fish track in relation to various wind farm features and operation, but also in comparison to the flow fields using the Moray Firth hydrodynamic model. I'd just like to say a big thank you to all the people who have helped support the study, and thank you for listening.
Hello everybody, my name is Michael Bevins Cameron. I'm a first year PhD student at the University of Essex, and today I'm going to be talking to you about using natural tags to trace salmon provenance, and my co-authors are listed here on the screen. So the focus of my PhD is all about tracing salmon provenance using natural tags. So we've seen a widespread decline in UK Atlantic salmon populations, and that makes it increasingly important that we understand their habitat needs and vulnerabilities. The declines are thought to be driven by mortality at sea, yet there remain large uncertainties about where different populations are going at sea. So the current most common method to ID salmon provenance uses genetic markers. However, many of the populations across the east coast of Scotland, as you can see from this map here, are genetically similar, making it impossible to track their marine migratory pathways at the river level. So thus I would like to assess whether it is possible to combine these genetic methods with the chemistry of archival tissues. So given their apparent vulnerability at sea, we would like to assess the potential for impact of offshore wind, as seen as there's a significant rise in developments off the east coast of Scotland, and that includes our industry partners, Kincardine Offshore Wind Farm Limited. Now, these wind farms could potentially have positive or negative impacts on salmon survival and migratory ability. So, for example, they may alter prey or predator abundance and or homing ability by increasing noise pollution. And for floating wind farms, the subsea cables and dynamic cables could potentially alter electromagnetic fields. Now, trawling and tagging studies have highlighted overlaps in post-smolt migration routes and the planned or ongoing development of wind farms along the east coast. As you can see from this map here, the grey areas indicate the development sites, the lines are for trawling areas, and the circles indicate salmon abundance. So we plan to analyse the archived post-smolts from these studies once we have the assignment methods dialed in. So the initial aim for my project is to build a chemical reference library of known origins for 7 key catchments across the East Coast genetic grouping, and this will identify whether catchments exhibit unique chemical fingerprints. We'll then use these knowns to train a variety of assignment models using different markers and tissues to identify the optimal approach that minimises cost and sample preparation time whilst increasing accuracy. So at this point, we will also test the potential for scale chemistry, as scales and fin clips can be taken non-lethally. So we will use these classification models to assign post-smolts that were caught at sea and hopefully to their catchment of origin, and use the information to identify population-specific migratory pathways to assess whether offshore wind farm developments could impact some populations more than others. We will also use these classification models to assign post-spawned adults caught in river to their catchment of origin to assess strain rates. And then this can be used as a baseline to infer whether future offshore wind farm development impacted homing ability. So for my PhD, I will be focusing on 7 rivers that are all in that East Coast genetic cluster, and nearly all of these are special areas of conservation. For the first part of my project, I will be analysing otoliths and eye lenses from kelp carcasses, and these were sampled by fishery trusts and boards from the annual salmon head sampling programme, and this was started in 2020 by my supervisors Anna Sturrock and Colin Ball. Now, the downside to training classification models with these is that we don't know their true origin and the sample sizes are low. On this map, I'm showing the 7 priority rivers and the planned juvenile sampling sites on the River Tweed. The background colours show the geologic strata. I'll be meeting with the various fishery boards and trusts in coming weeks to discuss collaboration and opportunities for sampling in summers 2024 and 2025. And these juveniles will form our reference library, and that will allow us to assess within versus between catchment variation, as well as to test stability through time. With the known origin fish, we will test different assignment methods using trace element concentrations and stable isotopes in the otoliths, eye lenses, and scales, alongside genetic markers such as SNPs. And by comparing different combinations of tissues and markers, I hope to identify the optimal natal classification approach using machine learning methods such as random forests. Otoliths and eye lenses, and also to a certain extent scales, they grow incrementally over a fish's lifetime, and that leaves us with a chemical chronology of what it ate and what water it grew in from birth to death. You can see a line of spots on the otolith here where I analysed this chemistry for my undergraduate project. The first part of the time series represents when it was living off the yolk of its mother, and then to when it emerged from the gravel and started feeding off of invertebrates in river. And this portion is the material we will use to analyse to assess natal origin. From here, the fish moved out from its naval tributary through to the estuary and then finally to sea. So a subsample of adults from UK populations and Irish populations were used to test whether natal portions of otoliths and eye lenses were able to successfully differentiate between populations, and this indicated significant differences between those populations and 77% classification accuracy using otoliths alone. So we expect this to increase by combining across tissues. However, it is important to remember that some of these adults may have strayed from other catchments and they were caught across a short period of time and limited geographic area. So in terms of research outcomes for this project, it offers the potential to differentiate genetically similar populations, and this could provide a better assessment of at-sea migration routes and highlight possible population differences in the level of impact from offshore wind farms. It will also provide unique methodological advances by combining tools for fish provenance and traceability studies that have previously not been combined, and the underlying classification methods will be useful across taxa. We also hope it will aid sustainable management of offshore wind development through providing a better understanding of stock-specific migration patterns. And for impacts, the research addresses a high priority evidence gap identified by the ScotMER programme in 2023 to understand spatial and temporal distribution of outmigrating smolts. So we'll be providing baseline data and new approaches for monitoring salmon migration pathways and their interactions with offshore developments. We also hope that this will help inform future policies associated with development of offshore wind farms. And finally, I'd just like to say thank you all very much for listening. And as you can see from the very long set of acknowledgements, it really does take a lot to get the project going. So I'd like to say thank you to all of our funders and everybody involved, and particularly those of you who have braved the elements to collect samples for us so far. Thank you very much.